Hello and welcome, everyone. I'm delighted you can all join us. This is the Christopher Sporborg annual lecture, which is part of the National Brain Appeal talk series. I'm Mike Hanna, I'm a neurologist and I'm the director of the Institute of Neurology. Today we'll be revealing the plans for the National Brain Appeal £7 million commitment to UCL's exciting new Translational Neuroscience Centre based at Gray's Inn Road, and that's what we'll be talking to you about and giving you an update. If you have any questions for the speakers today, there'll be a Q&A at the end of the lecture, moderated by Jackie Ashley, the chairperson of the NBA. Please post any questions in the Q&A section at the bottom of your screen, and we'll do our best to answer as many of the questions as we can. So you're all really welcome, and I'm going to kick off by sharing my screen. Okay, so I'm hoping that you can all see that screen. Unless someone tells me otherwise, I'll continue. What I'd like to do is just trying to forward my slide. So I'm just going to give an overview of the new building and the concept of translational neuroscience, which is where we're trying to take science towards patients to develop new therapies. And we believe that the fantastic environment that this new building will create will catalyze our ability to do this. So just briefly, the Institute of Neurology has around 1,200 staff and students, and it's arranged around 8 departments. And each department tries to link science by doing very basic science to understand mechanisms of disease right through to patients. And there's a bi-directional link where patients inform us about the type of questions we should be asking through our observations in patients. And then we do pursue these and look at mechanisms through laboratory science. And this link between patients and science is a fundamental feature of the work of Queen Square and has been ever since Queen Square began. There's currently around 300,000 patients coming to Queen Square every year, and this is a fantastic resource for partnership and for research. One example of this is our clinical trials facility in the hospital, and we have a large number of clinical trials happening here. And these include examples of things like new genetic therapies, where we're taking things into patients for the very first time in a highly controlled and careful environment. So one of the big advances which this new building will allow us to pursue much more actively is in relation to genetics. Interestingly, at least 10% of all neurological diseases turn out to be due to single gene problems, including conditions like motor neuron disease and Parkinson's. And there's been a transformation, as you may know, in discovering new genes and understanding the genetic basis of these diseases. And also what's happened is we have increasing ways to manipulate genes and replace faulty genes using things like viruses. And work is already ongoing in the Institute and is going to move into patients very soon, for example, in epilepsy and also in trying to treat spasticity, which is a big issue in many neurological diseases. And we are going to take these type of therapies into patients in this Wolfson trials unit that we have in the hospital. And we predict that actually many more genetic diseases will be potentially treatable by these sort of approaches over the next 5 to 10 years. And the new building is going to be really key in catalyzing examples of new therapies like genetic therapy. Like a lot of big projects, this began about 10 years ago when we thought we would try and build something in the goods yard behind Queen Square House. And that over time changed into this fantastic new opportunity to build a building on Gray's Inn Road. And initially we had no funding, but slowly and steadily we've managed to get closer and closer to our final target, which is 281 million. And the building will open in 2024. And here's an artist's impression of the building on Gray's Inn Road. It's about 7 minutes walk from Queen Square House, down Guildford Street, turn left onto Gray's Inn Road, and it's the old Eastman and Royal Free Hospital site. The site actually has 3 elements to it, and it's the green element in this diagram, which is the old Royal Free Hospital, which will be the new translational neuroscience building. The other parts of the site are also owned now by UCL, and they will also be redeveloped subsequently. Work is well underway. Here are just some initial pictures of the demolition. The demolition.
Mike, sorry, your slides are not showing.
What, the whole time, or?
Yes.
Oh, sorry. Okay. Right. So I can. Are they showing now?
Yes, that's great.
I apologize. I didn't know that. I didn't get a message. Yeah, I'll just go back a couple of slides. So the new building is illustrated here, and it's located on Gray's Inn Road, which is a short walk from Queen Square. It's about a 10-minute walk down Guildford Street and turn left. The site has got 3 elements, and it's the green element on this diagram. The old Royal Free Hospital site, which will become the new neuroscience building for the Institute of Neurology. And work is well underway. The demolition has commenced. Here are some images of the process, and actually the demolition has been completed. And it's going to be a really fantastic building which combines both patient services, outpatients, imaging for patients, as well as laboratory research, all in the same building, which is really, really important. There's 3 main partners in the building. So there's the Institute of Neurology, predominantly the laboratory research, what we call the wet lab research. And that includes colleagues from something called the Dementia Research Institute, which is a major national initiative for dementia research. It includes patient services, as I've mentioned, and also the headquarters of this National Dementia Research Initiative, all co-located in the new building. It's very important to point out that we will retain major presence at Queen Square. In fact, half our staff will remain at Queen Square from the Institute of Neurology's perspective, and roughly half will be in the new building. And we'll retain floors in Queen Square House, which is the big concrete building in the corner of Queen Square, where certain types of research where it's beneficial to be next to the hospital will continue. And as some of you may know, there are many buildings around Queen Square which are for research that doesn't involve laboratories, but more sort of office-based research and also imaging research, and that will continue at Queen Square. So we talk about this sort of dual hub where we have at Gray's Inn Road, predominantly from the Institute's perspective, the laboratories and what we call wet lab research, but also there'll be some patient services there. At the Queen Square end of the dual hub, that's where we would do clinical trials and more technically complex interventions on patients in this dedicated trials unit. And so we think there'll be a sort of flow of people and knowledge between the 2 centres. And this, of course, is all taking place in a very rich environment, a very rich neuroscience community across Bloomsbury, with some of the other collaborating centres illustrated here. So the labs will be state-of-the-art, open plan. We're looking to share a lot of what we call core technologies in the new building. And one of the very unique features actually is there's going to be a whole suite of highly complex, what we call core facilities, which will be new technologies, which will be useful to all the scientists and will enable linkages actually between patient tissues and patient materials that are donated with consent, for scientists to be able to study. And I'll just talk about a few of those very briefly. So one of them, which the next speaker will talk about in more detail, will be a dedicated stem cell facility. Stem cells, as you'll hear, are a very powerful tool for studying diseases by taking patient cells and then studying them in the laboratory. I mentioned earlier, when you couldn't see the slides, that gene therapy is going to be a big development for many neurological diseases, and there'll be a dedicated genetic therapy platform to help us in developing genetic therapies. And also, as you'll hear a bit later, I think from Tarek Yousry, there'll be a major MRI facility with a combination of 5 MRI scanners, including the most powerful example of MRI, a 7T scanner. So all of these facilities will be incredibly powerful in terms of advancing our translational research mission. Tarek will tell you more about the imaging. So this is going to be a really fantastic setup. It's actually unique nationally, and it's pretty unusual internationally. We're going to have dedicated core facilities. Scientists, clinicians, and patients will all be working in the same building in the best traditions of the Institute of Neurology in Queen Square to try and really take science to patients and develop therapies for patients. And there'll be this important sort of dual hub working in partnership between the Institute of Neurology, Queen Square, the hospital, and the new building at Gray's Inn Road. So it's a really fantastic opportunity, and we're incredibly grateful to the National Brain Appeal for the crucial support that NBA will give to making this finally a reality, which we think will catalyze progress to develop therapies for patients. So I apologize that my first few slides were missing. I wasn't aware of that, but I'm now delighted to hand over to my colleague, Professor Selina Wray, Professor of Neuroscience at the Institute of Neurology, who's going to talk a little bit more about the stem cell technology, which is so powerful. Thanks very much, Selina. I'll stop sharing.
Oh, thank you to Professor Hanna for the introduction, and thank you for the invitation to speak to you tonight. I will actually speak on behalf of both myself and Professor Ricky Patani, who will be here at the end for the Q&A. We're very excited to be part of this new Translational Neuroscience Centre. And actually today I will share with you our vision for something that's been an ambition of ours for a while, which is to develop a state-of-the-art stem cell facility for the modelling of neurodegenerative disease. And I just check that I can advance my slides. There we go. So to just give you an overview of what I will speak about for the next 10 minutes or so, I first of all want to introduce the concept of stem cells and why they're so crucial to our understanding of neurological diseases. I then want to illustrate why the Institute of Neurology is really the natural home for this sort of work. And to do that, I will give just one example of how we've modelled Alzheimer's disease using stem cells, and really use that to illustrate why this is such a powerful technique. And then finally, I will finish by speaking about the new facility and how it will really enable an expansion of this work, and why it's such a natural fit with the rest of the Institute. So if we start off with just a little bit of signposting to really provide context for these models and why they're so important, I would describe myself as a molecular neuroscientist, which I think is quite typical of a lot of scientists, that we have fancy ways of describing things that are really quite simple. All that molecular neuroscience means is that we're really focused on understanding the small stuff. And the reason we want to do that is we really believe that understanding things at the molecular level is the key to unlocking our understanding of disease and using that to develop new treatments. So we already heard from Prof. Hanna about our expertise at the Institute in understanding the genetic basis for neurological disease. And it's really the genes and the proteins that are produced from those genes that make the smallest building blocks that go on to form the cells of our body. These come together to form our organs, such as the brain, which then come together to form us. And the reason I showed this schematic is because I think it really gives context that if something goes wrong at this very, very small level, the impact of that is felt at each increasing level of complexity. And so if we want to develop effective treatments for neurological disease, we really need to be targeting the disease at this very small level. Now, that sounds simple enough, but of course, when we think about diseases that affect the human brain or the human central nervous system, things become more complicated. So what we typically do when we're working at the molecular level is when we have a gene that we know causes disease, we use that to build models so we can understand the consequence of those genetic changes. So that could be a mouse model, it could be a fruit fly model, or it could be a cellular model, so cells that we grow in the dish. Now, what the challenge is for when we're looking at diseases that affect the human brain is it's really quite inaccessible to us during life. So we can study the brain at post-mortem, but of course here we're looking at the end stage of disease. We can look at the brain in life using non-invasive imaging techniques. But if we really want to see the changes that are happening in brain cells, we need a way that we can culture these cells, grow these cells in the laboratory. So we can really use our molecular techniques to understand what's happening at the cellular level. Now, our field has really been transformed in the last 10 years through the availability of stem cells to model disease. Now, most people, I'm sure, will have heard of stem cells in one context or another, but just so we're all on the same page, I thought I would share a little definition. And when we talk about stem cells, we're really talking about two key properties. The first one is self-renewal, which means a stem cell will divide to form two identical stem cells, each of which will divide to form two identical stem cells, and so on and so forth. And the second defining property of a stem cell is this idea of pluripotency. So this is derived from the Latin from most potential, and it means that a stem cell has within it the ability to form any cell type of the human body. They're essentially a blank slate. This includes neurons and other brain cells. And what has really been transformative in our field is that for many, many years, it was thought that this was a one-way road. So a stem cell would become a specialized cell, but that process couldn't be reversed. We now know that this isn't the case, thanks to some Nobel Prize winning work from 2012 that showed we could actually take a stem cell, a specialized cell type such as a skin cell or a fibroblast, and we could reset the identity of that cell and take it back to a point when the cell is unspecialized. We call these induced pluripotent stem cells because we're inducing the property of pluripotency. And the reason that this has been so transformative is it means that we can now derive patient-derived personalized models for neurological disease. So we can take a patient or a research participant who has a disease of interest with a known genetic cause of that disease. This, of course, is a unique strength of the Institute of Neurology, is our strong patient cohorts. And we can ask them if they would be willing to donate a skin biopsy, which allows us to grow their skin cells. This is non-invasive. It's a non-invasive procedure for the patient, and skin cells are very easy for us to grow in the lab. And we can then use these skin cells and reset their identity so that they become stem cells again. And then by using specific factors in the lab, we can change the stem cells into the cell type that is affected in disease. So in the majority of our diseases, we are converting the stem cells into brain cells or neurons. And because the patient's genetic information is conserved throughout this process, this means we have a patient-specific model of disease that we can use in the lab to really understand the very earliest things that are going wrong within these cells. So to just share an example of this that I think is really powerful, because it's all been centered around the Institute of Neurology, the National Hospital for Neurology and Neurosurgery. My own group works on our understanding of Alzheimer's disease. And this is the first family where a genetic cause of Alzheimer's disease was identified in 1992. So the majority of Alzheimer's disease is not genetic in the sense of being passed from parent to child, but there's a very, very rare number of cases, about 1% of the total number of Alzheimer cases, where it is in fact genetic. And John Hardy and Martin Rosser, both of whom are based at UCL, were able to identify the cause of the Alzheimer's disease within this family. Now, what did that allow us to do? Well, it was a breakthrough right from the beginning, because it allowed us to start to put together a sequence of events. So we know in the Alzheimer's brain, there are two main changes that occur. There is a buildup of a pathology known as plaques and a second pathology that are known as tau tangles. And all of the genes that have been identified are associated with the development of plaque pathology. So this tells us that plaques happen first and tau happens later. But we've really struggled to understand the full sequence of events that goes from the initiation of plaques through to neuronal death and the manifestation of clinical symptoms. And part of that, the reason for this is that mouse models do not capture this whole process. There's something very human specific about the susceptibility of brain cells to these Alzheimer pathologies. So what we've been able to do in the lab since this technology became available is to grow cells from some participants who have these genetic causes of Alzheimer's disease. And what you can see on screen here is an image of what these brain cells look like when we grow them in the lab. I think they're really quite beautiful. And you can see these green long processes, which are very typical of the shape of a neuron. This is what we call an axon, and they're really key for the cells to be able to communicate with one another in the brain. And that brings me on to my next slide, which is to say that for us, it's not enough for the cells to just look like a brain cell. They need to act like a brain cell as well. And what you can see here is the cells that we grow in a dish are sending signals to one another. That's every time a little cell, one of the cells flashes, it's sending a signal, it's communicating to the other cells in a dish. And this allows us to now take cells from individuals with no disease-associated gene together with Alzheimer's cells, and we can look for differences between these two cell populations in the lab. And we can try to understand the very first things that are going wrong in the Alzheimer's cells with a view to stopping that from happening. This is my one slide of data that I won't go into in depth, but just to show you that even growing these cells just for a month or two in the lab, we start to see disease-associated signatures in our Alzheimer's disease cells, which are shown here, that are not present in the control cells. And of course, the strength of our institute and this translational approach is from these same individuals, we have detailed clinical information, we have matched samples such as CSF or spinal cord fluid, and we also have brain tissue that has been donated at post-mortem. So we're really taking this kind of holistic approach to understanding this person's disease. Now, we haven't only done this for Alzheimer's disease, that's just a case study, but actually the same principles that I just described can be applied to any neurological disease of interest. And I just highlighted some of the papers here where we've looked at motor neuron disease using this approach. We've looked at Parkinson's disease, we've looked at frontotemporal dementia. I'm using the royal we here, this is across the institute, and actually many of the papers that I'm showing have been led by Professor Ricky Pitani, who will join us for the Q&A. And what this really shows is that already we've been early adopters of this transformative technology, but it's still somewhat restricted to people who have the specialized know-how and the specialized technical skill set to work with these cells. And so what we want to do is change that. We want to really be able to harness the power we have in terms of excellent patient cohorts to enable any group who is interested to use this technology to really develop a bespoke understanding of disease. And this is our vision for a new facility. We want to enable and expand and accelerate disease modeling by making a purpose-built facility rather than what we have currently, which is a facility that's retrofitted to the existing building. And we want to provide training and work with other groups who are interested in using this technology to model their disease of interest, but perhaps don't have the manpower or the resource to build this from scratch. And what I really like about this approach and why I think it's the natural home to have this at the Translational Neuroscience Centre is it's really bridging the disconnect that we've perhaps had to contend with as molecular biologists before, where we'd have a whole range of research happening in the patients, but we would always have to look to a mouse model or to a generalized non-specific cell line. We don't have to do that anymore. We can really use personalized bespoke models to gain insights into pathology mechanisms and also for the development of new drugs. And I think by using these human models that are patient specific, the new insights we will get from these models will really increase the chances of successful translation back into clinic, which of course is core to the mission of all of us who are here today. So this just summarizes what I've said. You know, one of the challenges we've had to overcome when we think about understanding disease and developing new treatments is the fact that the brain is both complex and inaccessible. But we're now able to gain insights thanks to these new advances of cell models. And we can be optimistic that our advances in this area will really allow us to have a better understanding and enhance our chances of successfully developing treatments which make it through into clinic. And we're both very excited to lead this platform, and really we can't wait to get started. So thank you all for your support. And I will finish here and stop sharing. Ricky and I will take questions at the end. But in the meantime, I will hand over to Professor Tarek Yousry, who will speak to us about the MRI scanning platform. Thank you.
Yes, thank you very much, Selina. Wonderful presentation, wonderful pictures. I'll just start sharing now. Okay, so in terms of the imaging, and Professor Hanna has previously briefly mentioned the MR facility, I would like to mention first our current department in the hospital at the National. So we have within the department 4 scanners, a 1.5 Tesla scanner, that's the field strength, which is relevant, which was installed in 2008. It's currently to be replaced. We have a 3T Tesla scanner from 2010. We have an interventional scanner, which was actually very much supported by the National Brain Appeal, and we're very grateful for that. I'll mention that at the end again. And then a research scanner, which was acquired in 2008 and was upgraded in 2015-16. So this is what we really have within the department, and it's really mainly 2 scanners. Which is a tremendous shortfall in terms of our imaging capacity. And this is in the context of actually the whole country, because the Royal College of Radiologists has made a survey some years ago in which it showed that UK is really within the European countries quite at the bottom, both in terms of the imaging capacity, the MR scanners that are around, and also in terms of radiologists being available to report images. We are below Turkey. So clearly a very severe shortfall in terms of capacity. How do we deal with that? Well, within the current situation, of course, we try to extend the working hours, and in that we use all 4 scanners, which gives us the equivalent of an additional scanner. And we got an additional mobile, which is outside at Queen Square, and we are trying to outsource as much as we can. But with the outsourcing, all of our colleagues, which is very nice to hear, want their own patients to be scanned at Queen Square, not to be outsourced and reported by us, but that's of course not possible, otherwise we wouldn't be outsourcing. So there's clearly a need for additional imaging capacity at Queen Square. And this is what we are looking at. So the new building at Gray's Inn Road, which will have within it as a core facility an MR suite, which will be, this is the picture of it in terms of the layout. So the blue is what's supposed to be research scanners from UCL. The green is what would be the NHS. So 6 scanners in total, of which at least 1 will be 7 Tesla and the rest will be 3 Tesla scanners. This will be run as a single unified facility. And I think that is very important, that we have a very flexible usage planned. So this will, on the one hand, increase the capacity to quite an important degree and certainly serve our patients for some time. The other important advantage is that the integration with research will help to provide a better management of the patient, better diagnosis. We will have a single procurement of everything, UCL, UCLH. We have unified financial arrangements, so we don't have to scribble around who pays for this or that. It will be the same approach, irrespective of where the patient will be scanned. And the scanning ability, the kind of sequences will be the same on all scanners. But is it only a question of capacity? Of course, the way I ask the question, it's not. There's also the issue about quality, the kind of scanners we're getting. Here it is quite important to look now at the field strength. The 3 Tesla is very important. Most of the district general hospital will have 1.5 Tesla, which is perfectly sensible. We also will have one and will remain having one, which is perfectly for many diagnoses and many questions our referrers have. However, the 3 Tesla does give us an edge, which is critical as we are in one way tertiary or a quaternary referral center. So really the last address the patient can go to. So we need to be able to address rare diseases or difficult diseases. The integration with research will enable us to adopt changes as quickly as possible. And the fact that we can use the same scanners both for research and for patient care is supporting that approach. And just a few examples of the challenges that we have at the moment and in the future. A very simple thing, the contrast that we're using based on gadolinium, which is supposed to be quite a rare material, a rare earth, and it has been used since the late 80s. It's widely used. We like to use it. Why do we like to use it? Because it is safe. It has been used 30 million times. The side effects are minimal, are much less than any kind of other contrast in CT, for example. So we really use it very much. Excellent safety profile. However, about 8-9 years ago, we started to notice that there are some actually gadolinium diseases. One of them is so-called nephrogenic systemic fibrosis, which happens with certain kind of contrast in a certain circumstance where the patient may have problems with the kidneys. And that was a new disease, which has also disappeared because, for instance, the European Medical Agency has said that certain contrasts should not be used, so were eliminated from clinical usage. And we need to take some measures, which we took here quite rigorously at Queen Square, to look at the function of the kidney, and since then that disease has disappeared. But it just shows you it was safe, but under certain circumstances, things can become complicated. The other thing that we notice is, that if you give this contrast in a short period of time, a number of times, you can see some deposition of it in the brain. Now, interestingly, it doesn't seem to have any effect on the patients, whether it is about thinking or movement, cognition, or any kind of motor expressions. It doesn't occur. We don't know whether it can have something on the long run, but again, the EMA decides, European Medicinal Agency, that certain contrasts should not be used, and we are taking appropriate measures. But clearly, there is another problem, which actually is quite serious, which is the environment, the pollution of the environment, which I have to admit was not high on our horizon of radiologists, but it is a problem. It usually doesn't occur. You have difficulties in finding it, but now we find gadolinium in the rivers, in the coasts, and in some of the materials that we eat. So it's not a surprise that we need to do something about it. And that's just one of the examples that were shown. These are embryos of sea urchins. And there are different ones. Now, I never saw, as a radiologist, I have to admit, this was not my main field of interest. But if you look at these different kinds, these are the normal ones, these are abnormalities of the embryos of this kind, and of that kind. Now, they do happen, as they always can happen, that things can go wrong. The point is, how do they change? Is there any relationship with they were exposed to gadolinium and to an increasing amount of gadolinium? We know that gadolinium doesn't do anything to us in the contrast form. That means, just very briefly, you have the gadolinium, which is quite toxic, but it has a protective shield around it, a chelate, which ensures that it doesn't go out, it doesn't have any effect on us humans. But when it goes out here, you can see that this is good, black is bad, and you see that with the increasing amount of gadolinium, you get more and more malformations of those embryos. It's different for different kind of embryos, but there's clearly effects on those creatures. So we must do something to try to control how much of this gadolinium goes out in the environment. And we have a critical role as neuroradiologists in doing that. Now, in terms, just 2 examples or 3 examples how we can do that. ASL is a certain technique, arterial spin labeling. It's a certain technique by which we can see the flow of the blood through the brain. It doesn't need contrast. Here you have a patient who has a tumor of the brain in this area. We give contrast. We didn't see any enhancement. Enhancement is not very good. So here is a few months later. Again, not much of enhancement. You look at these images, you don't see a difference. However, if you use other techniques which do not require contrast, you see that in this area, actually, there is a certain signal. This is a certain technique called diffusion weighted imaging. And this is the ASL, where you can see how the blood goes through this area. And you see there has been a change over the months, but we don't see the change here. So we can play a trick with the enhanced images and subtract non-enhanced from enhanced and see whether we see anything. Well, we don't see anything here. We might see a little bit of lighter signal here. It's a little bit whiter, but that's very subtle. That's not subtle. You don't need to be a neuroradiologist to see that that's shouldn't be there. So that would be one way of assessing this change without using contrast. This cannot be done at 1.5 Tesla. This can only be done at 3 Tesla. Another example. We know now, since 2016, there has been a change in how tumors are classified, and the genetic classification is key and is getting more and more important. Now, imaging is very good, MR is very good, is very sensitive in showing anything that shouldn't be there, in showing any pathology. But it's not very good in telling us which kind exactly of pathology it is. So here we have 2 tumors, look quite similar to each other in some ways, but if I apply a special technique, which is called spectroscopy, I can see that one of them has these double curves. They are usually not there, but if I have them, it is a specific kind of tumor which is treated in a specific way and is usually a better outcome. So this would provide me with something close to histology. It's almost like the Holy Grail. It is exceptions, but we are a big tumor center at the National. We have a lot of research happening here. And if you think a biopsy, if the patient doesn't want to be radiated, we can only do a biopsy, we'll only see a certain area within the brain. But if we can do the spectroscopy, this kind of technique, which is a challenge, but we can do that, we can differentiate these two kinds of tumors. And it has an important effect on the patient. Again, this cannot be done at 1.5 Tesla. This can only be done at 3 Tesla. We have a lot of innovations at UCL and at UCLH, which we're doing. So how does it work? We start somewhere in the preclinical phase. This can be a phantom, it can be animal testing. Quite often it is phantom. And then we have the so-called first-in-man. That's the first time we use it in a volunteer, and then we try to validate it, we explore it, we use it in a single center, multi-center, and so on. But until we get into the clinical into the clinical world and apply it on patients, we have quite a significant attrition. Not many things don't go through here. And that's something we need to address, and we need to make it quicker. So from a clinical point of view, we have a problem with the capacity. We have also a problem with, in certain areas, that we as neuroradiologists, although we are very good with what's called eyeballing, we're quite sensitive. But certain areas, we are not that sensitive. We are not very good, and we know that. So there's another question, how can we address this? So we established something that we called 5 years ago, so-called quantitative initiative, which we tried to use quantification as a tool. For instance, in dementia on a tumor, is there really a change in volume, or in MS? If I have 25 lesions, do I see number 26 and 27? How can I do that in a way that always provides you with the same kind of an answer? Again, it is at 3T that we can provide computational support best. And this is something we're very proud of. It's collaboration. Do you call it collaboration when it is within one team? It is the team around Professor Duncan and ours and others in the Faculty of Engineering, who looked at, in epilepsy patients, there's a certain structure, the hippocampus, which is read here, and it changes in volume and gives a different signal. But how good are we in seeing that? And how can I do that in an objective way? So now I can get, just with a push of a button, a few hours later, I admit, but still with a push of a button, I'll get this kind of information for every patient who I think there might be a problem with this structure. And I can see immediately whether the volume, as it is here on this case, is too low or not. And I can make comparison with the two sides. I also can see the change in signal. So this is now used within our NHS imaging service. And it's really a tremendous step forward. And we hope there will be more to come. And that's the next thing that we hope to come, is with patients with Alzheimer's disease, where we know there might be, so this is the one side of the brain, the other side of the brain, and then you parcellate it into what we call lobes, and then smaller areas within the lobes. And then you look, how is this volume compared to a population that you know are not affected by dementia? So this is a volunteer not affected by dementia. You see certain areas are a bit less, but not really of worry. This is a patient with Alzheimer, and you see clearly the red where you have lost quite a lot of volume. And what we're trying to do is, before we get here, to get to this stage where we can see, okay, so something is going down, we are losing volume, we need to get in and try to treat. This is a bit late because it's difficult to regrow, at least at the current stage. Now, another thing is, from a computational point of view, what's called deep learning. Again, with tumors, you can work, have a specific analysis of images, again, without contrast, where you can get the information I showed you a moment ago about with the spectroscopy, just by computing those images. Now, again, this is not in the clinical arena yet, but there is potential for that. So this new facility will clearly address the shortage of capacity that we have. It will accelerate, help accelerating the translation of research into the clinical practice. and will clearly help improving diagnosis and patient management. And in this, I must say, the Brain Appeal, the National Brain Appeal, was over the last 20 years helping at quite crucial time points. Some may not remember that in 2004, 2005, 2006, it was through the Brain Appeal that we got our first PACS, at least at the national. It was the initiative that led the whole trust. It was from the Brain Appeal, the support that came, that allowed us to do that. Also, what I showed you a moment ago, the interventional MR, being able to operate, being able to image patient in theater, was, again, we were the first in the country to be able to do that. And again, here, I think we will be put by this into a leading position in translating innovation into patient care at a certain level. So with that, I would like to thank you. And again, there will be questions later on. I would like to introduce now Professor John Duncan, who I had mentioned earlier already. John. I'll stop sharing, of course.
Thank you, Tarek. Right, I'm going to try and share my screen. Good. Can you firstly hear me and see the screen? And thirdly, can you see the pointer?
Yes.
Yes, we can see it.
Okay, all good, 3. So I'm going to talk about the way we're going to integrate research and all the fantastic things we've been hearing about from my colleagues with the clinical practice, and how we're going to increase the involvement of our patients in our research projects. I've been a clinical academic neurologist at Queen Square, dare I say, for over 30 years now, and I've seen huge advances over this time. And this is going to take us another quantum leap forward into our research, and what is most important to me, the application of this to our patients for their benefit. I'm going to describe to you the current problem, the solution, the context of the new clinics that are going to be built, how these will work with clinical research, and lastly, how you can help. The problem is that Queen Square is full up. Every year we see 190,000 outpatients, and 46,000 of these are being seen for the very first time. Our clinics are very crowded, and this is enhanced, as you would imagine, with the need for social distancing in the waiting rooms and in the clinics. Of course, we do more by video and by telephone than ever before. Indeed, telemedicine has really come on 10 years as a result of the coronavirus pandemic. But for many new patients, people who are seeing for the very first time, people we have yet to build a rapport with, people in whom there needs to be a difficult conversation, and to be able to see how the patient and their family are reacting to what the doctor is saying, difficult decisions need to be really with a face-to-face consultation. So there's a huge need for more clinic space. The other point, which actually surprised me, but it is the fact, is actually only 3% of our current patients participate in research, 1 in 30. But also, Queen Square will need more inpatient capacity over the next 3 years. More surgical wards, more operating theatres, more critical care, and high dependency. And this is why the dual hub that Mike Hanna was alluding to earlier is so important. So the solution is to create the dual hub of integrated clinical neuroscience and research at Grayson Road and at Queen Square. So we're integrating clinical neuroscience and research, and also integrating across 2 sites, Grayson Road and Queen Square, just about 7 minutes walk apart. Now, the way this is going to be divided up is that ambulatory care and outpatients and the fantastic MRI that Tarek was describing just now will be focused at Grayson Road, whereas at Queen Square, the focus will be more inpatients, surgery, and critical care. And staff and patients will go to both sites during their times. Now, the context, again, to reiterate the important point that Mike made earlier, is that the co-location of clinical work and research in the same building is of vital importance, vital both for patients, relatives, and for researchers. Patients and their families coming to Queen Square, often with very difficult situations, very difficult conditions, tertiary or quaternary referrals from around the country, find it very uplifting to know that in the very same building, there are researchers dedicating their lives to their condition, say dementia, of course, but also other conditions, stroke, epilepsy, MS, brain tumors, movement disorders, neurodegeneration, motor neuron disease, long COVID, autoimmune disorders, spinal cord troubles, nerve and muscle disorders. The list goes on and on. Further, the researchers can meet with those individuals who are affected by the conditions that they are researching. This is hugely motivating for researchers, and it also helps to ensure that the direction that their research is taking is relevant to the patient's conditions, and they aren't just going down some arcane alley that's really of no clinical relevance. Thirdly, this allows public patient engagement, so that the patients, the relatives can talk with clinicians, can talk with researchers, and really engage in understanding each other's perspectives. And this is going to be key in the patient research hub. So, to get your eye in, this is the front building on Grayson Road. This building here seen from the other side. This is where the site has been cleared. And Tarek and the MRI units will be in level -1 in this sort of area here. And above that, on the ground floor, will be the new outpatient facility. This is on the ground floor, so Grayson Road is along here. There's entrance through here, a courtyard, entrance into the main building, and outlined in red is the new outpatient center. This will have automated check-in. Patients will come in through here, check in like in a modern airport. There will be research displays saying what research is ongoing at the moment and how patients and their relatives may be able to take part in a research project. There'll be 22 clinic rooms, a pharmacy, mouth swabs for taking DNA samples, the facility to take bloods and do minor procedures like cardiograms or skin biopsies to produce the precursors of stem cells, as we heard earlier. This is the ground floor of the facility. This is the courtyard that we spoke about earlier. And the patients coming in from Grayson Road will come through this refurbished old building into this courtyard and then come through here into the outpatient clinic, which is over to the left-hand side. There will be a welcoming environment, very different to the kind of cramped, crowded clinics where we currently work. And there'll be a comfortable, welcoming environment for patients to have in-depth consultations with their physician, their surgeon, their rehabilitationist, their speech or language therapist, physiotherapist. Now, at the moment, only 3% of our patients are involved in research projects. I think the main reason for this is that there, at present, is a grave lack of space. There's no privacy, there's no physical resources for patient and public engagement. There's no ability or space to explain research projects and findings. There's nowhere where an in-depth, confidential conversation can be taken to obtain informed consent. Does an individual really want to take part in research, but never want to push that on anybody? There's nowhere where research blood tests can be taken out, where ECGs or mouth swabs or skin biopsies can be taken. At the moment, if you go into a clinic, research enrollment is course further constrained by the coronavirus. Even before that time, you would see research fellows standing in the corridor talking to patients, a really very unsatisfactory situation and location, and really not surprising that only 1 person in 30 would participate in research. So our goal with this patient research hub in the outpatient facility on the ground floor will be to increase participation tenfold from 3% to 13%. And this will take 2 broad areas. Firstly, specific research projects. For example, there will be specific clinics for motor neuron disease, for multiple sclerosis, for stroke, for epilepsy, for dementia, of course. And these clinics will be concentrations of individuals with those problems. And of course, the researchers working in those areas will know when those are and can participate and meet the individuals concerned. Secondly, there will be generic, more big data research, whereby anybody coming to a hospital for any reason, and also their relatives, their friends, who may act as normal controls for comparative studies, for clinical research, MRI, blood work, and of course, for analysis of their genes. In the patient research hub, this will be integrated within the outpatient clinic. There will be electronic displays in the foyer and in the waiting rooms and the corridors of the latest research finding relevant to those studies in those conditions that are being treated and being assessed in the clinic going on at that time. So, for example, if there is an epilepsy clinic on a Wednesday morning, the latest epilepsy research would major in the waiting rooms, as well as the generic big data researchers. There will be research ambassadors for generic research and for the specific diseases on hand to explain and answer questions. There will be private rooms for detailed explanations, for the obtaining of informed consent, and for other investigations such as clinical examination, tests of cognitive function, blood testing, and mouth swabs, and so on. So, how can you help? Well, you can help the National Brain Appeal to help our patients. As a rough estimate, the clinic, half of the ground floor, cost about £10 million to construct and to fit out, and individual 20-metre square clinic room will cost about 260,000, and a 12-metre clinic room will cost about 156,000. So thank you for your interest. Thank you for helping the Brain Appeal help our patients. It gives me great pleasure to now introduce our chairperson, Jackie Ashley.
Thank you very much indeed, John, and thank you all so much, all of you who've come along this evening to explain how our wonderful new facility is going to work. I think it's a hugely exciting development, and as some of our speakers have said, it really is absolutely groundbreaking. It seems obvious, doesn't it? We bring together the researchers, the scientists, the clinicians with the patients, but it's just not done at the moment, and hopefully this will set a whole new standard in the way we do research. So it's very exciting to be a small part of this. We at the National Brain Appeal have pledged to raise £7 million. We'd like to raise quite a lot more, but anyway, 7 million is what we have said so far. And what our 7 million is going to go towards is 3 different areas: the stem cell facility that we've heard about, the MRI scanning unit with 6 of these state-of-the-art new scanners, which I think again is a very exciting development, and I was particularly pleased to hear the environmental concerns there as well. That was great. And finally, the thing that I think is the most important really, which is the patient research hub, because I think that's going to make all the difference. It's quite shocking really that only 3% of patients at the moment are contributing, and I think even 30% is maybe a bit of a low bar. Let's hope it'll be more than that, but it's all too obvious how much having patients taking part in the research, and it just becomes a normal part of their hospital experience, I think, will hugely benefit the research project. Now, to make this work, to get more money in there, we obviously need help. And that's where I'm going to put in a little appeal to those of you in our audience who, many of you have already contributed generously, but perhaps you could dig deep, you could ask your friends, you could look around and see what you can do to help us some more. We're looking to help nearly 15 million patients in England alone who suffer from neurological disease or neurodegenerative disease as well. So it's a big number. 1 in 6 people in our country will at some point suffer from some kind of brain disease. So probably one in your family, who knows, or certainly a friend or a neighbour. It's going to affect all of us. It's a very big thing that we need to play our part in. Now, if you are able to donate, please visit our website, nationalbrainappeal.org, and select the New Home for Neuroscience Fund. We have a variety of projects, but this particular one is the New Home for Neuroscience. So if you are able to donate some more or something new, then thank you very much indeed. As you can see, it will be very welcome. Now, we're going to take some questions. We've got all our professors here. We've also got Ricky Batani joining us, who will also take questions. So I'm thinking people are going to be putting them in the Q&A for me, and I'm going to start off by asking one from Melody McLaren. Unless Melody, are you here? Do you want to ask the question yourself? Maybe not. Maybe I'll just ask it on her behalf. So Melody says, will the scaling up of patient participation in research be supported by the network of NIHR research champions working with the research ambassadors, as well as by physical facilities? John, I think that's probably one for you. John?
Yeah. Yeah. Is that better? I'm sorry, Jackie, I'm afraid you turned me off.
I'm so sorry. How did I do that?
I can't imagine. Well, the answer is yes. NIHR is key actually to this, and speaking as one of their senior investigators, I know very well how important the involvement of NIHR is. And in addition, complementary to that, the LCRN, or the local clinical research networks, will are fundamental in providing funds for recruiters and people who will interest patients in studies. I think this is absolutely essential, because the clinicians have to get on and do the clinical work, but having roving ambassadors who know about their areas can engage in conversation, and particularly, find out who's interested in what. Often, in a particular area, in a particular morning, there probably will be a total of 10 or 15 studies that somebody could participate in, and different sorts of studies will resonate with different people. So yes, NIHR funding is absolutely key to this.
Great, thank you. Now, I've got a question from Adrienne Banks, who I think is about to leave, so we'll take it quickly, who is asking what funding is coming from the government, if any? John, that's probably one for you again, I would think.
Well, as you know, the NHS produces funds the kind of standard clinical provision. Of course, funding is tight, we all know that, and particularly in the last 2 years, there's been a huge call on the exchequer. I think we don't seek the National Brain Appeal to support what the NHS should be paying for anyway. What we seek to do is we seek to raise the bar to do more than the NHS standard provision, and also to do it first. And there are many examples, Tarek alluded to several of those, whereby the National Brain Appeal allows us the National Hospital at Queen Square to take the first steps and show the improvements that can be garnered from this. And in organizational terms, correct me if I'm wrong, but I don't believe that there's any center anywhere that has a dedicated patient research hub built into the hardware and the culture of an outpatient clinic.
Yeah, and that's what's so exciting. We've a question now from Frank Curtis. Will the need for research using animals fade away altogether in time? Interesting question. Who would like to take that one?
I'm happy to start. So hi, everyone. Thanks very much for joining today and for your ongoing support. I mean, animal models, I think it's important to start by saying that they've been hugely valuable across a range of basic and applied neuroscience. I think it is important that we do seek to reduce, refine, and replace animal models, and some of the approaches that Professor Selina Wray was talking about, these human stem cell approaches will indeed help us do that. But I think it would be perhaps short-sighted to say that, for me to say that they would be altogether replaced, because at some stage, I think showing that a particular mechanism of disease is relevant in a sort of living organism is really key. But I think it's a really insightful and important question, and it's our absolute duty to reduce animal usage and suffering as far as possible. But I think we do that by complementing them with these newer approaches rather than aiming to replace them altogether, which I think would weaken our portfolio.
Did you want to come in, Selina?
I would absolutely agree with that. And I think one of the things that I perhaps didn't highlight in my talk when I showed all the different models is I'd place them together like a jigsaw, with the idea that they're all complementary, but they all come with their strengths and their disadvantages. I would also add to Ricky's point that I think animal models are particularly important when we think about drug screening, because although we can try drugs directly on the cells in culture, what we really need to know is how that drug is metabolized by a whole organism too, in case there are any harmful byproducts that may be formed by other organs. So yeah, it's our hope to reduce and refine, as Ricky said, but we won't be able to replace altogether just yet.
Thank you. Now a question from Wray Harris. What new abilities will the 7T scanner provide to the researchers? Tarek, that sounds like that's one for you.
Yes, of course. So the 7T is actually a completely different beast. You can see details of the cortex, of the gray matter of the upper layer of the brain in a detail that can get quite close to microscopy. I think it's a completely different level. I think there's more work happening now, so that kind of 7 Tesla is not only used for research, only not to reduce the relevance of that, but also to expand its need for specific indications for certain diseases. One is, again, epilepsy. We know that we have a number of patients with epilepsy where we don't see anything on imaging. And we think it has to do with the resolution of the current scanners, and going to the 7T will just enable that and will add that level of detail. If you think of completely different disease, movement disorders, where patients, either are very inhibited in their movement, facial expressions in Parkinson, or if they have a tremor and movement, you can do something called DBS, deep brain stimulation, by which you have to target specific structure in the brain, which is being done at the moment. But for instance, I know from Amsterdam that they have that opportunity to use 7 Tesla images, which is a completely different. So it's not like 1.5 to 3. It's a completely different level of detail that you can see, and it would allow then a better targeting of structure that are difficult to target at the moment. So I don't expect that 7Ts will be everywhere, but I do expect that we will be starting using them in a clinical environment, and that other centers will follow us.
Fantastic, thank you. Selina, a question for you about stem cells. How do you get the samples, and how do you grow them, and how long does it take? Very sort of basic layperson's question.
So I think maybe Ricky and Prof. Patani and I could both comment on this, because I think we both have some different perspectives. So in terms of actually getting the samples, the thing that really makes this possible for us is the co-location with the clinics. And the reason I say that is because, as I mentioned, we take a skin biopsy and they are perishable. We need to process them ASAP once that sample has been taken. So actually the advantage of being where we are now is when samples are taken, we normally have them from clinic to the culture dish within a matter of hours. So this is only at the skin cell stage. Now, from the skin cells to derive the other cell types we work with takes a while. So it takes about 3 months to develop a stem cell line from the skin cells. And then when we take those stem cells and use them to make other cell types, it really depends on the cell type of interest. So we are making cortical neurons. So these are the neurons that are affected in Alzheimer's disease. And that takes us several months. It takes between 3 and 6 months to make the cells and mature them in the lab. Now, the reason I said that both Prof. Patani and I would kind of comment on this is one of the things in the field has been, can we shorten this timescale? Can we make things quicker? And I know that Professor Patani has done some really breakthrough work in this area. So I might hand over to you now, Ricky, to comment on the motor neurons.
Thanks very much. So, I agree with everything that's been said so far. And actually, you know, our labs work very closely together, and we discuss this quite frequently. And I think that both viewpoints are really valid, and it's great that we can sort of coexist and discuss these things. So what I would say is that the length of the actual protocol to differentiate these stem cells or to transform them into the particular cell types that we want is to some degree less important than making sure we actually get the right cell type. I think that's the key, and I think that this attribute, this property of pluripotency, which is this a state where you have master stem cells that can become anything in the body. I think the key is really understanding how to sort of guide them into becoming the particular cell type that you want, because the possibilities are limitless. They could become any cell type in the human body. So this, I think it's useful just to try and explain it through an analogy of schooling. So it's like having a very a bright 10-year-old coming to you and saying that, you know, I want to be an astronaut. So what does this person need? Well, you need to make sure that the child has potential and that they're given particular pieces of advice at particular stages. And I think that analogy is actually quite a powerful one for the stem cells. So we know that they have tons of potential, but what they need is stage-specific education, if you like, and actually the only reason we're able to guide the cells to become these clinically relevant populations is because of the mouse developmental biology that's come before. So again, it just really highlights how intertwined these things are, and you know, we should aim to, just relating back to the last question, we should aim to refine and reduce animal usage, but we can only do that because of the usage that's happened thus far.
Brilliant, thank you. If anyone else wants to ask a question, we've still got a couple of minutes left. Do put something on the Q&A for me. But meanwhile, I have another question for John Duncan. John, you're obviously going to be getting a lot more research, hopefully, once things are up and running. Is the plan to share the research internationally as well?
Absolutely. I think my colleagues will give their views, but I think certainly the areas I'm working on, we're moving towards open science, whereby we say on a platform like we use one called GitHub, for example, where all our data and how we did it actually is available. So somebody in Montreal, Barcelona, Sydney, Tokyo can access it as we can. I think that's fundamental to move forward. I think the concept of scientists being secretive about what they're doing and keeping it to themselves really belongs in the last century.
Yeah, yeah. Tarek, did you want to come in on that? You're muted, Tarek. You need to unmute.
Thank you. So it's absolutely, as John said, so there are two levels, I think. One level is what we always have been doing, which is communicating our findings. And that happens in international meetings and presentations, papers, and collaborations and so on. But we all realize that actually being in silos is not of advantage to anyone, even for the individual researchers. It's much better to collaborate, get one's work acknowledged, embedded in others' work, and then accelerating. I think we recognize that that was something that happened certainly over the last 20 years, that that we are trying to build as big networks as possible and as sensible, not always as big, beautiful, but where it makes sense to leapfrog, to accelerate, to be quicker. And that's throughout. So for instance, we are trying to work since some time, the example that I gave, with the Faculty of Engineering. And we, even before COVID, we were able to communicate and collaborate with multiple sites, whether within the country or outside.
Well, Tarek, I suppose another follow-up question from the international question is, if someone, I don't know, say in Canada or India, had a very rare condition and would benefit from the new super scanners that you will have, would you do the scans for them? And obviously you can just send them presumably by email. Is that something you would do?
Well, I think if that was the case, then of course, yes. It's rarely so clear, you see.
Yeah.
Jackie, it's rarely so clear that we have one thing not, you know, there was the example when there was a proton beam treatment and we didn't have it here in the UK, and they went to the Czech Republic. In imaging, if we had such a situation, certainly we will be accepting anyone and try to make that happen.
Wonderful, thank you. Right, I have time for one or two more questions. Someone has a hand up. John.
I just wanted to say that this building is designed to be environmentally friendly, and we're well aware of the need to curtail global warming, and this building has been designed exactly with that in mind, to minimize the carbon footprint and the amount of waste and the effluent that is produced, both from the research and from the clinical activities that we do.
Great to hear. Right, well, I think if there are no more questions, I'm going to say a huge thank you to everybody. Thank you to our professors, Mike Hanna, Selina Wray, Richie Batani, Tarek Yousry and John Duncan. And for all of you for joining us this evening. I think we've had way over 60 people joining us on this webinar, which is fantastic. And hopefully this time next year we will be in person. We live in hope. But if you've any questions at all or want to find out any more about this wonderful new project, do just get in touch via the web page, the website, or indeed with Anna, who deserves a special word of thanks, Anna McLeod, who has organised this event, and also thanks to Teresa, our wonderful CEO at the MBA. So without more ado, I will say thank you so much for joining us. Enjoy the football, those of you who are going to watch it. I personally am not, but I'm sure a lot of you are. It's been a great evening and I feel very excited to have heard a bit more about the wonderful plans that we have. So we look forward to hearing more in the future. Thank you very much and good night.